just going to go over some preliminary work that we conducted as part of the most recent lobster benchmark stock assessment, looking at shell disease, drivers, trends, and trying to understand how this data can be used for management guidance. For the story of shell disease, it's often intricately tied to the history or the stock trajectories for southern New England lobster. And just as background, when looking at the spawning stock biomass for lobster through time, for southern New England, what we've seen is, or what we saw was a stark increase from the 1980s and 1990s towards its time series peak in the late 90s. But thereafter, we saw a precipitous decline in spawning stock biomass through the 90s and much of the 2000s to where we're at now, at time series low for the population. And of course, fishing harvest has been attributed to what we've seen in that decline in the 2000s, but in a rapidly changing southern New England environment, there's been a number of other stressors for southern New England lobster. Some of the big ones being increased predation, whether it be striped bass, spiny dogfish, or black sea bass, a changing thermal environment with increasing temperatures in many estuaries and coastal waters like Narragansett Bay, around perhaps not providing suitable thermal habitat for lobsters anymore. And there's been a number of different anthropogenic stressors, such as oil spills or chemical infections to chemical contaminants in certain areas of the stock's estuaries, like this example here from the North Cape oil spill in 1996. However, the one aspect that's unique to lobsters specifically is shell disease, which is going to be the basis of my talk today. So epizootic shell disease or ESD is a bacterial disease which often is represented by the breakdown or the degeneration of the lobster shell or the cuticle. And so here we have two examples here of a healthy lobster on the left versus a diseased lobster on the right. You can just see how the carapace is often highlighted by this rotting of the shell. It was first observed in 1996, so towards the peak of the spawning stock biomass for the southern New England stock, and first observed in Rhode Island both by state scientists and the industry. And at that time, it was so novel to see such a disease pervasive within lobsters that the data collection for this became pervasive across federal and state monitoring programs, whether it be for fisheries, independent or dependent sampling, with some of the trajectories here just showing the increase in prevalence that we saw from 1996 when it was very low, or before that zero, towards prevalences that could reach as high as 40 to 45% prevalent in lobsters depending on the location and season. Why do we care about shell disease? Well, the major concern is that it's a new aspect for natural mortality for the stock, in that it seems to prevent lobsters from successfully molting and ultimately cause mortality. So in this theoretical model, looking at health on the y-axis and successive molt cycles for lobster, you can see that during the molt, if health were to decrease, particularly with enhanced shell disease, bold here, as health declines, yes, you can technically molt out of a shell disease carapace to improved health, but not necessarily to a health that you once were prior to your molt. And with successive molts, you might continuously lose health to a point of mortality. And then more quantitatively, in some monitoring programs like in Connecticut from the Millstone Lab, researchers have found that roughly 30 to 45% of the lobsters with disease survive compared to those without disease. And probably one of the largest challenges with shell disease is we don't actually know what causes it. We just know what correlates with its presence. And so we've seen a number of different anthropogenic and environmental correlates with shell disease. One of the principal ones being temperature, with lab experiments showing that an increasing coverage of the shell has been associated with warming waters, whether you're in a light kind of coverage state or a moderate coverage state for shell disease. And there's been work that's try and then understand how shell disease correlates with temperatures through time, and whether looking at indices such as just average sea temperature or days above a certain stressor temperature like 20 degrees Celsius or 20.5 degrees Celsius is indicated here. Although, as you can tell, the correlation through time looks somewhat strong since the disease has been prevalent, but not prior to its emergence. And so what we've seen mostly for shell diseases in southern New England, which, as people know, has been a primarily low state for the population. However, what we've now started to see is an increase in shell disease prevalence in northern regions. And this kind of latitudinal gradient of stronger disease prevalence in southern areas compared to northern has been found on a smaller scale, such as in Massachusetts waters, but in areas that traditionally had very little or no shell disease, we're now seeing greater prevalence through time. And so you can see that from sea sampling data from Maine DMR, looking at how through their lobster management zones from G through A here, you can see the same latitudinal effect, as well as within years, you can see that prevalence has really increased in the 2000s or 2010s. So in a rapidly warming environment like the Gulf of Maine, while the levels are still low for diseased lobsters, there is concern about what this might mean as a future stressor for this highly commercial valuable stock, as well as a new source of natural mortality for this population. Historically, shell disease data has been used in two facets for lobster stock assessments and management. The first is that each time we conduct a benchmark assessment, we provide not only results from the assessment model, but also model-free indicators, and this gives board managers a bit of a more observational and less quantitatively rigorous option or tool to try and understand stock trajectories and concerns that may then inform management decisions. And so particularly those for managers that are worried about perhaps an assessment's uncertainty or retrospective pattern. As well, we have done a number of sensitivity runs and analyses of trying to see how any type of shell disease time series could be used to inform natural mortality within model parameterization. So this is on the right one example here from the 2020 benchmark assessment, where instead of parameterizing natural mortality as just constant through time, there's actually a time series informing natural mortality from shell disease data of a long-term monitoring survey. The current challenges that we face for this work is that when we do either the indicators or kind of this model informing, or this trend informing the assessment model, there hasn't been any standardized surveys across stock units that have allowed for direct comparison. Oftentimes, while we all measure disease the same now since 1996 in terms of classifying it, there hasn't been the exact same surveys standardized that allow for an easy comparison. That's been one of the criticisms from reviewers of previous shell disease work in trying to looking at this at a bigger scale. So the purpose for this work was to try and evaluate shell disease data from one standardized survey that 9 minutes both stocks to inform the trends and severity of loss. And so for that, we used the coastwide ventless trap survey that's conducted in both southern New England and the Gulf of Maine. It's in partnership with industry that uses pots to sample lobsters across their wide range of habitats. The pots are ventless, so they don't have vents, and this focuses in on the juvenile portion of lobster stocks within state waters. But it does follow a random stratified sampling design with the trends from these data sets used within stock assessments as relative abundance indices. We collect a lot of different data on lobsters. They're very unique, and with data streams being able to inform all different types of science and management questions for the populations. The data stream that I'm primarily focusing on today is shell disease. And as I mentioned earlier, we do have standardized methods for characterizing shell disease, not just presence or absence, but then severity, with some examples here of what we would call light coverage, moderate coverage, and heavy coverage for shell disease. For the modeling work that we looked at for this, we used generalized additive models for lobsters and trying to look at just the presence or absence of shell disease, or the probability of a lobster carrying shell disease. For now, what we've done is basically modeled it as binary, given with codes 2 and 3, so moderate or severe disease being the presence of shell disease, and 0 and 1 being absence. And the 1 being considered absence is to guard against perhaps observation error of someone thinking it's light shell disease, but it's actually damaged shell or other biological growth for someone that might be newer to monitoring disease. And we've looked at data from New York up through Maine in this, and modeled shell disease separately for both stocks, given differences in minimum sizes and things that can affect the actual results from the GAMs. And just as some of the questions that we were trying to think through here, besides just temporal aspects of shell diseases, how much does the prevalence vary by males, females, or females carrying eggs? Do we see some of the latitudinal or zonal gradient effects with disease as reported previously, and trying to capture the nonlinear aspects of shell disease with size? And so just briefly looking at some of the work for southern New England, and kind of highlighting each of these figures really quick, we see that disease is greatest in females carrying eggs, and this highlights both the larger individuals molting less, as particularly those with eggs molting less frequently, given the taking a year off from molting when carrying eggs. And we see that the disease prevalence increases as you get bigger, highlighting that smaller lobsters tend to molt more frequently and thus able to shed disease before it becomes too prevalent. The seasonality of the disease is well captured within southern New England, where it's highest in June, right before the molt, and as lobsters begin to molt and shed their exoskeleton, the disease prevalence decreases for it then to build up subsequently thereafter. And we see kind of a zonal effect from the data in southern New England, where 611 or Long Island Sound had less disease prevalence than Rhode Island Sound, Block Island Sound, and Narragansett Bay, with the highest prevalence in the Buzzards Bay area in Massachusetts. And some slight or perhaps significant trends in prevalence from the year effects of the model. We see a lot of the same effects here for the Gulf of Maine. Female eggers have the greatest prevalence, a little bit less of a strong contrast with size. With disease prevalence, we don't see that same cyclical molting pattern here. We actually see a slight decrease into October from June. But again, the same latitudinal effect is reported previously, except now extending down through Massachusetts and New Hampshire waters, where we see that the greatest prevalence is more towards Massachusetts Bay, Cape Cod Bay, with decreasing prevalence towards down East Maine. And so from these models, we can then start to try and think about producing standardized indices through time, see how shell disease has actually changed. So now better accounting for varying factors and experimental design. So as examples here on the right, we have indices for Gulf of Maine and southern New England for shell disease of female eggers at 83 millimeters in June for 2 different stat areas. So ideally, we would like to use some of this information as updated inputs for Model 3 indicators, but there's some things that we'd like to look at further in terms of both the model structure for how we want to model shell disease. Do we want to look at presence absence, or model individual classifications, such as light, moderate, or severe, and as well as some of the other covariates. Temperature is obviously one that's is obvious in terms of an environmental driver, but other ones such as depth, stratum, that better integrate the sampling design of the surveys. And with that, I want to first thank all the many field scientists and industry collaborators that we've had to complete this work. Without them, it wouldn't be possible, and the various state and federal funding agencies that have helped. And if there's time, I'll take it.